Okay, so let me just see if I'm on my thing. Yes, I am. Okay, so now we're going to talk about session 8, which is the primary visual cortex. So you remember when we did the LGN, right, Jeanette?
Mm-hmm.
Okay, so, this is just basically completing the circuit. And I always say that, like, we go to the back of the, we go all the way through the visual pathway. And this is like, we like leave the country, so to speak. And then we turn around and we return back to the country. So next one will be back in the retina. But this one right now, we're going to talk about the primary visual cortex because it's important, because you should know these things. So again, remember what we're talking about and why we're doing all of this. We're doing all of this so that way, at some future point, you can actually read an OCT and know what's in it and like how it's connected. And I think we're doing a good job of that. But you want to know, like, why are we doing all of this, right? The whole pathway doesn't just terminate once you get to the LGN, it goes somewhere else. And again, this is what we're looking at. I always say all this stuff, I just always repeat this probably because I didn't take out this slide. But the thing is, just remember that if you don't know what's going on, it's okay because it's very complicated. Okay. We're trying to make it simple, but it's still always going to be complicated. Never get frustrated. So we're going to go through this again. I always show this, and just so that way we hit it really strongly. So this is darkness. So when we shine light on the central photoreceptor, remind me, is that active or inactive?
On the central one, then it's inactive.
Correct. And then therefore, is glutamate secreted or not?
Not secreted.
Correct. And then which bipolar cell is activated, on center or off center?
The on center is activated.
Correct. So the on center is activated. And then the horizontal cells that are connected to the central photoreceptor, is that horizontal cell active or inactive in this state?
Active.
They're inactivated.
Inactive. I did that last time too.
Inactive. Yeah, and what the way I remember it is that I remember that they're inactivated because the central photoreceptor and the horizontal cells are like at the same party. And the whole point is, this is like your buddy when you're throwing a party. They're going to, of course, like and be the same as you. They're on your team. But then they go to the surrounding houses or the surrounding photoreceptors, and they do the opposite of what we're doing, so we get better signal to noise ratio. So what happens to the surrounding photoreceptors? Are they active or inactive in this state?
The ones that are currently inactive?
Yeah. What happens to them?
Oh, then those are then active.
Yes, they're inverted, right? So the whole point is so that when you look at the central photoreceptor, the valence or the polarization is going to be so different, right, than the surrounding, that we know what's going on in the central photoreceptor. Does that make sense?
Yes.
Okay, so that's that's your lateral inhibition paradigm. Okay, then the bipolar on cell, who does that cell activate? The ganglion on or ganglion off?
The ganglion on.
Correct, because they're in the same IPL, remember? So they're in that sublamina B, you remember? And so you have that same hand holding, it's got to be in the same spot. Okay, and then the ganglion cell sends its axons to where, generally speaking?
To the LGN.
To the LGN, right? And of course, there's the amacrine cells to the LGN. Okay, and then we're going to look now. So now we're at the LGN, right? And then I say, okay, it's ganglion cell to nerve fiber layer to optic nerve to optic chiasm, and then we remember the contralateral nasal retina, right, comes in, and the ipsilateral nasal retina goes out, and then you get this through the optic tract to that ipsilateral LGN. You remember that?
Yes.
Right, at the decussation. So you're getting the fibers of the contralateral nasal retina to come in, and then you're sending away your nasal retina to the other side. And that's why you're getting those at the LGN. And if you remember, when we talked about the blind spot, that was the reason why the blind spot was sort of showing you that. You remember this?
Yeah. Yeah.
Okay. So then you remember that these are the 3 pathways that actually go through this particular set of structures, right? I mean, the other ones do too, but they don't go all the way to the LGN, right? So there's 5 classes, but only 3 of them actually go through this. You have your midget ganglion cells, which we know is also called beta, which we also know is the parvocellular pathway, or the P type ganglion cell that goes through the P cells or the parvocells in the LGN. Then you have your parasol ganglion cells. Remember, red, green, and they're called alpha because they're a little bit bigger. And that's the magnocellular pathway. The magnocellular for motion. And then you have your bistratified ganglion cells, or gamma as we call it, the coniocellular pathway, because conio, I guess, means dust. So they're smaller and they're defined by their the size in the LGN. And that's the one that does blue, proprioception, kind of like the add-on pack. You remember all this?
Yes.
And then there were those two other classes. One was the intrinsically photosensitive retinal ganglion cells. They had melanopsin inside of them. Their classical example that I like to say is the giant retinal ganglion cell. They're very rare, but they deal with kind of like your ambient conditions, your pupillary light response, blah, blah, blah. They can respond to photons themselves. You remember this? And then it's like Edinger-Westphal complex and suprachiasmatic nucleus and ventrolateral preoptic nucleus, all those things that I was talking about, right? But they're not the LGN, right? And then you have your other, which is like your superior colliculus, which deals with saccades, SCCS, superior colliculus saccades, right? And again, they're not going through the LGN per se. They're not synapsing there, right? So you remember all of that, right? Those 5 classes?
Yeah.
Good. So, we want to talk more about this though, right? So here's our LGN. It doesn't stop there, right? They're synapsing, but they're not ending there, right? So if we remember, here's our LGN. It's at the thalamus. And then again, we're looking at our cross section here. And remember what I said, 2, 3, and 5 were 23 high five, I guess. It's like Michael Jordan high fives you because he's on your side. He's on your team, ipsilateral. And then 1, 4, and 6 are contralateral. You remember this, correct?
Yes.
Good. So then we look at it again, and I'm just doing that for us. I don't know. I'm rehashing this for you. Ipsilateral temporal retina, right? You remember this, and then where does it go? Well, it goes somewhere else, and it's going to go to the primary visual cortex, okay, or as we call it, V1. And the primary visual cortex gets something from the LGN via the optic radiations. Okay, so that's what we call the pathway from the LGN to the primary visual cortex. And so here is your kind of optic radiation pathway. And we're going to show you, right, this lateral geniculate nucleus from this this ventral surface of the brain. And you can see them very clearly here. And I'm showing you where the LGN is. There's the LGN. And then you have those optic radiations. Okay, and then if you go from this particular side, it's easier in a sagittal section to kind of look at it. And so here you have these 2 ways that you get the optic radiation. So this one is basically going through the parietal lobe. It's kind of going like upwards, if you will. And I want to just point out, and I'm not sure, I think this is from your book, but I don't remember. But if it is, I believe it's from your book. But notice what they say here. They say fibers representing the superior retinal quadrants or the inferior visual field. One thing I know it's like easy to remember, but then it's also easy to mess up, is the fact that when we say fibers representing the superior retinal quadrants, that's very confusing because we're talking about retinal quadrants. We all have been taught about the inferior visual field. It's like the opposite. Do you remember that? It's like go by the visual field nomenclature, less so the retinal quadrant nomenclature. Make sense?
Yeah.
Because otherwise you're going to confuse yourself. And I remember when I read this, I was like, oh, wait, what? I'm confused. I always thought it was like inferior equals superior and superior equals inferior, right? And then you're like, wait, no, they mean retinal quadrants, which itself is reversed, right?
Yeah.
Okay, so then you remember that the other one goes through the temporal lobe, and we call these this like the Myers loop. You remember that?
Mm-hmm.
Okay, good. And then where do they end? They end in the occipital lobe, which makes sense, okay? So just remember, they're talking about retinal quadrants, so they write that, but it's better to look at visual field quadrants. Okay, and then where do they end up, right? When we look at our visual field, I think this is directly from your book. It's kind of like, inverted a little bit again, right? So here's your visual field, right? And you look, here's the central one. Well, the central visual field fibers actually end up in the posterior non-central part of the brain, right? And then your superior visual field fibers end up in the inferior part of the brain. And then your inferior visual field fibers end up in the kind of like the superior part of the brain. And then your peripheral visual field fibers are actually in the center part of the brain. Make sense?
Yeah.
It's just all like inverted, but relative to the visual field. Okay, that's all I'm trying to emphasize here. I know that you've learned it in medical school, but it doesn't hurt to just keep hammering away because it's very important because we deal with visual fields, right? Okay, so, and then, yeah, I'm showing you superior, I'm showing you inferior, and I'm showing you how central, how they're on the opposite sides, okay? All right, great. So now we say, okay, well, once we're in the V1, where do we go? And I'm not going to get too much into this, but it doesn't even end there, but all the interpretation of the stuff, right? We could go to a V2, a V3, then there's a V5, and it's like a V4, and all these other things happen to make post-visual processing occur. And, just one of the things I say is, it's like the ventral surface is the what pathway, and the posterior and the dorsal surface is the where pathway. It's kind of like two different ways of understanding your visual phenomenon, okay? So that's how you kind of go. If you can remember that, that's helpful. But remember, we also call our favorite part of the brain, which is V1 again, the striate cortex. And there's a reason why we call it the striate cortex. And we'll get into that. And it's very important because it actually is a very, very special anatomic designation. Okay, so it's the striate cortex. Striate just means stripe. Okay, you see a stripe. And what does that refer to? We'll talk about it, okay? So now we know this goes to V1, right? It doesn't matter where I synapsed here, I'm going to V1. It just depends on where in V1, but I'm still going to V1. All right, so now that we're here and we go through the LGN and then we synapse and we're going to V1, we say to ourselves, well, what is V1? And I'm actually drawing here this kind of square, okay? And this is actually the primary visual cortex. If we like cut it kind of sagittally and we looked at like the thickness of its the actual surface itself. So like this is like the top of the brain, like the dorsal surface of the brain, and this is like the ventral surface of the brain, and we're cutting the visual cortex almost like we were looking at the thickness of the visual cortex of V1. And notice what they've done. They've from outside, so this is the surface of the brain to the in of the visual cortex, we number it. Imagine there's a 1, there's a 2, there's a 3A, a 3B, a 4A, a 4B, a 4C alpha, 4C beta. Now, this is where knowing beta and alpha actually is very helpful. Remember I told you that the midget ganglion cell, which is the smaller cell that synapses with the parvocellular pathway or the parvocellular cells, they're also called beta, correct? Well, now this is going to pay off a very small, but I think a very important dividend, okay? If you look, these guys end up in 4C. So what happens is it's not just V1, okay? It's not just V1. It's actually one of these 4C alpha or 4C beta, okay? And so because this was a parasol, you may not remember, but this was a parasol cell, which means that it was actually synapsing in either 1 or 2. This parasol cell was going through the LGN. It's an M-type parasol ganglion cell. When it's moving, the M-type, and notice how they write P and M here, but they mean the type from the LGN, the P and the M. So now you know this is coming. This M is where the parasol cell has synapsed with it. And then that M-type goes and it synapses in 4C alpha. So the alpha ganglion cell terminates in the alpha layer of 4C. Does that make sense?
Yes.
And that's just, know that that's just somehow how it works, okay? And then of course, if we say, and those are called the alpha cells. So it actually kind of helps us if we know this like non-standard, but visual scientists use this as a term, nomenclature. So then let's look at our little midget cell, right? Here's our midget, right? We know this because this is the dendritic connection. So this would be like the IPL. It has a little kind of dendritic tree. So it is a P-type midget ganglion cell. Well, what is a P-type midget ganglion cell? Who do they synapse with? Well, they synapse with the parvocellular cells or the P cells of the LGN. Okay, well, the P cells of the LGN, I also know this guy's also known as beta. Well, he's going to come and he's going to synapse in the 4C beta part of the primary visual cortex. So 4C alpha and 4C beta all get 90% of the input. Remember, it's like, 80% are midget ganglion cells, 10% to 12% are, parasol ganglion cells. So 92%, let's say, of our ganglion cells terminate in 4C alpha and 4C beta. And not only do they terminate in incredibly structured ways. You have the alpha guys, the alpha cells terminating in 4C alpha. You have the beta guys terminating in 4C beta. There's a reason why I'm doing this, okay? But I'm just trying to square away that it's mainly in 4C. By the way, notice who I didn't talk about. I did not talk about the coniocellular pathway yet. Okay, so we'll talk about that later. But 4C is what you want to remember of V1, and 4C beta is the kind of like the secondary layer of 4C. The beta layer is where the beta ganglion cell terminates its pathway. Makes sense?
Yeah.
Okay, so beta cells. All right. So, and then, I just want to also point out, because there's something very elegant about this pass-through situation. Remember, when we first come in here, look at this, it's like CIICIC, right? It's exactly what we said, contralateral, contralateral, ipsilateral, ipsilateral, contralateral, ipsilateral, contralateral, right? So it's all of these things, right? But then notice how they all form up into one ipsilateral column and one contralateral column. Mind you, they're first synapsing in layer 4, as I said, of what we call area 17, which is another name for V1, but they then reassemble themselves into contralateral, ipsilateral dominance columns, orientation columns in your head. Does that make sense? So it's, in other words, there's a structural kind of assembly that occurs back here in the occipital lobe. Make sense?
Yeah.
Cool. So, and I just want to show, right? Contralaterals, all these guys, ipsilaterals, all these guys. Okay, good. Okay. And then we call them orientation columns. And then if you put them all together, it's a hypercolumn. I don't know why they call it that. It's a hypercolumn. Okay. Okay. Well, now let's talk about our other friend. Now you already know, what this one is, right? Because I just drew it for you. There's 2 strata. This is a bistratified ganglion cell that is of the coniocellular pathway. And so through the LGN, it's the dust cells. It also goes to V1, but it doesn't actually go to layer 4, either, 4C alpha and beta. You and I already know 4C alpha and beta, those are the alpha and beta ganglion cells. It didn't say gamma over here. And sadly, they don't call this stuff gamma. But what they say is that these guys actually go and they connect into 2 and 3. And then this is the surface. And they synapse, and I kid you not, they synapse with something called blobs. I'm really not, that's literally what they call them, okay? And it's it's in 2 and 3. So you know how from the day one, I kept talking about how this is like an add-on pack, how it's like, this is like if you're playing a video game and it's like you need motion and you need red, green, and that's like, you can go through life that way. But then you got this add-on pack where it's like, I can see blue and I can do proprioception and all this, putting together all this different stuff and processing and stuff. Well, that's like an add-on pack. Well, look, if this is almost like this is the brain and it's like we only have this and then we just kind of click this right on like an add-on pack, okay? And so it's in 2 and 3, and if numbered starts at 1 from the outside going in, then 2 and 3 is just under that. And so we say layer 2 and 3 is where they are, and it's blobs because these stain with something called cytochrome oxidase fairly well. Like you see a blob, like it's a discrete blob. And so this here is a neuron of sorts or some kind of synapse, but they stain really well. And so they're literally synapse with the cytochrome oxidase blobs in 2 and 3. Make sense?
Yes.
Okay. And remember, nothing has changed. We're still in the primary visual cortex. It's not like we're in like V2. Okay. And this is not like V4. Okay. It's still V1, 2 and 3 layers, 2 and 3 within V1, layer 4C alpha within V1. Okay. So that's what that means. Okay. So now, and then remember, we talked about it. You already know it's like 1 and 4 are contralateral retina. And look at this. So this is a book that like I just pulled this from somewhere, but now you know this is true. Non-P, non-M ganglion cells. Makes sense, right? Right? Non-P, non-M ganglion cells. I would argue that before all of this, you'd probably be like, I don't even necessarily know how to parse that. But now it's like non-P, non-M. That makes sense. It would not be a P and an M unless it was synapsing in 4C alpha or beta, because I know it's alpha, it's beta and alpha cells, right? And then here they're doing ipsilateral retina 2 and 3. Yeah, okay. And our convention is 2, 3, and 5, and 1, 4, and 6. For coniocellular cell layered cells, but 1 and 4 are contralateral retina, you know that, and 2 and 3 are ipsilateral retina, you know that, right? So this makes sense even if it's coming from a completely different book. Okay, so now we're gonna just put it all together. This is that same book. Okay, we just looked at this one over here, the coniocellular pathway, but let's go back this way, which is kind of where we're talking about. Here's your M-type ganglion cell. Now remember, M-type ganglion cell means parasol cell, right? M-type. So that means that, they say from layer 1 and layer 2. That makes sense because there's a magnocell, that's the magnocellular layer, an M-type ganglion cell. Well, that's an alpha cell. So where are they going to synapse? Well, guess what? 4C alpha makes sense. And then they have some post-processing stuff that happens, fine, but that's just very consistent. Let's go over to here. Here's your contralateral retina, P-type ganglion cell. Okay, P-type means parvocellular pathway, midget ganglion cell, red-green, okay, 4-6, makes sense. That would be the contralateral parvocellular layer. It's going to synapse in 4C beta, but let's follow this up. It's going to also go all the way up after going here and kind of meeting up with the blob too. And then there's literally something called the interblob. Okay. It's called the interblob. But doesn't it kind of make sense that you would need some kind of interaction with these cytochrome oxidase blobs because this is blue and you're dealing with red green? Right? It makes sense. That's what I'm trying to get at. Like now you look at it, it's not just memorization. This makes sense that you would have some kind of, handoff or, talking to each other situation here between these particular cells and these particular cells, the corneocellular cells, because they deal with color and it makes sense. Okay. So, and by the way, again, completely different book, right? Not our book, but now we get it. Right now we understand the visual science behind it. So that's why I like doing these. Okay. And then I'd like to point out that there's something called an interval. I don't know why I highlighted that, but there you go. Okay. That is a scientific term. Okay. And here's all this. Remember, this is layer 4, right? So layer 4 and particular 4C alpha and beta, there is a 4B, but notice that's for this post-processing stuff. But layer 4 is the key layer. Now, why do I keep harping on this? Yes, because 92%, okay, of all of our ganglion cells kind of synapse in that end up there in the pathway. But let's go ahead and look at this. Okay, this was a long time ago, a guy who's Italian, Gennari. He was a med student, if I'm remembering correctly, I believe so. He did this sectioning and he said, well, if I look at all of these things, I don't see much. And then all of a sudden you see the beginning of a line. And then he called this the striate area because look, there's a stripe, you can literally follow it. It's so perfectly aligned. What do you think that line is? Well, guess what? That line is layer 4. Okay, so layer 4 is the stripe. And layer 4 is literally so perfectly structured that at a bird's eye view, you can see the neurons aligning perfectly, because if they didn't, we would not have the structure of our visual field that we have. It has to be so precise that you can see it from a macroscopic view. Every single neuron is lined up correctly. I think this is fascinating. Okay, that's what the stride area is. The second thing I'd like to point out, let's say we were in 4C beta from here, and I could pick out, remember what I told you, the central vision is all the way over here in like the most posterior segment. There is a possibility that you could literally pick a spot here and it would connect backwards to the LGN, go backwards to a ganglion cell, a midget ganglion cell, go backward all the way to the retina, to one bipolar cell, and all the way to a single cone. You can have literally a single cone represented right back here, which I think is even more fascinating, that you could potentially do these types of moves, that you could literally send a jolt and go back and forth. That level of precision, I will point out, I know we're being recorded here, but I don't believe that exists for any other structure, organ in the body, that kind of precision. Okay. So that's why this is, it's really important, but that's why it's super cool, and why probably a lot of Nobel Prizes were won on the basis of the visual system because of how precise it is. Okay. So just remember, the line of Gennari is what an anatomist calls layer 4. Now, I didn't say layer 4 is only 4C. It's layer 4A, 4B, 4C, but it's layer 4 because the vast majority of our finished kind of, our ganglion cells terminate to create the striation. Got it?
Yeah.
Cool. Okay, so now here's another kind of, line thing from a book. Okay, and it's a little bit more complicated, but it's like, I just want you guys to be able to look at anything and say, well, I can maybe navigate this, even though it's not written perfectly like I want it to be written. So here's something you can already tell me. Okay, well, I can see here, this is probably a visual cortex V1, 2 and 3, there's some circles here. I'm going to say those are blobs, right? You know how I like doing this, right? Like I like just grabbing something and be like, okay, what do we know about it? Okay, and then I'm going to draw this line, right? Because this, for some reason, they have a 4C center. Okay, fine, 4C center. Well, let's draw a line through the center. Here's alpha and here's beta, right?
Yep.
And then you say, okay, well, there, is on this side, closer to the surface of the brain is alpha, and then down, and then that's 4C alpha, and then 4C beta is closer to the to the inner part of the brain. And so one of the reasons why I like doing this is so you know directionality. Again, the numbering nomenclature 1, 2, 3, 4A, 4B, 4C alpha starts from where? It starts from the dorsal surface, like the surface of the brain. It's as if you took a needle and went through the brain, you would be labeling the number, you'd be numbering, and the nomenclature would go in that direction, okay? And I like doing this because that way you can keep it in your head. Like, just like, when you go from, in the IPL, we talked about this, and you start numbering through the IPL, and where, sublamina A is. Sublamina A is closer to the bipolar cell, sort of near to where the original photoreceptor kind of transmission started. Sublamina B is further out. So we're labeling in that direction, okay? So in this case, we're labeling in the direction from the outer surface of the brain surface all the way down to the inner surface, okay? And so again, this is layer 4. That's the line of Gennari. That's layer 4 of the striate cortex, V1, area 17 from Brodmann's area, okay? I'm just throwing it out there. They're all like kind of still the same thing. Okay, and then we look here and it says, okay, here's the P, right? So they say P here, right? And so if it's P, we know if it says LGN, we know this means P type, which means midget ganglion cell, which means that it's gonna terminate in 4C beta. And then here they show that it also has some center connections. Okay, great. And then we say P and K kind of have a termination where they're in 4 alpha and they kind of overlay each other because K has to talk to them. But then here we go back to 4C beta and center, and then A. And all I'm doing here is showing you that there's a little bit more of a complication. It's more nuanced, but why I'm pointing it out is just that, again, the P cells and the K cells are marrying each other, so to speak, right? In 4A, or they're talk, and then they talk probably after that into this area and these blobs, and they do so. It makes sense that those two would be married to each other in some way. Okay. And then in the same way, in the center over here, right, you see P and M, of course, they're going to have some kind of marriage together because our midget and our parasol are the most frequent. And we still need to have a connection between our two most frequent, our bi-party system in our brain, so to speak. We want to have that too. Okay. And so this right here is the midget ganglion cell parvocellular pathway. These guys right here, which is alpha and center, are the parasol ganglion cells, also alpha, magnocellular pathway. And then your Ks are the add-on pack, right, where they have 1, 2, and 3, but mainly 2 and 3, where the cytochrome oxidase blobs are. And then, of course, 4A, where they're trying to talk with our red-green helper friends, okay? And so that's your bistratified ganglion cells or coniocellular pathway. Okay, so now another photo here. I'm going to ask you questions, okay, about kind of, I'm going to just point, and you're going to tell me what pathway, what ganglion cell, what is the information being conveyed, and then and I'm just literally pointing out something in the visual cortex. Okay, so I'm pointing to there. If I showed you something terminated there and I told you follow that neuron back, what is the pathway?
Probably the coniocellular.
You got it. And what is the ganglion cell look like?
It is bistratified.
And what does it do?
It does blue light and proprioception.
Proprioception processing. You got it. Okay. And I'm drawing a line here. Okay. What about this one? It's terminating in this part of 4C.
Maybe that's the parvocellular pathway.
So if it's the parvocellular pathway, what would be the kind of the alpha beta designation?
It would be a beta.
That would be a beta. And is this where beta would be? Would it be on top of 4C or would it be on bottom? Right? So that's why I was saying the nomenclature, if I was going to define it, it's like 1, 2, 3, A, B. So this would probably be alpha and this would probably be beta. Does that make sense?
Yeah.
So if it's alpha, then who terminates in 4C alpha?
The parasol.
Parasol, which is what type?
Magnocellular.
Magnocellular or M type. And so what does that deal with?
Motion.
You got it, right?
Yeah.
How about there?
Okay, so that's under, so that would be beta.
Correct.
Where the parvocellular line terminates.
Correct. What type of ganglion cell?
The midget type ganglion cell.
Midget type ganglion cell. And what does it carry?
Information about red-green light.
Red-green, you got it. Okay. Again, up there, tell me again what that is. So if it's 1, the add-on pack, right?
Okay, so it's probably a conio.
You got it. That's the guess, right? And I mean, it makes sense, right? Cornea cellular pathway, tell me again, it's what type of ganglion cell? Not to belabor the point.
Unstratified.
Bistratified, and what is it carried?
Information about blue light and
Blue
proprioception.
light and proprioception. You got it. Okay. Okay. And then what about there? So that's four. What would you say this was? 4A or B, first of all? It says A, B here.
That one may be A.
Okay. So there, I'm going to give you a hint. There are two of them that could be A. Okay. 4A. So if you had to kind of figure out which one would be, which one would get married kind of way up here, just see what is up here. And so who would terminate in 2 and 3? Just give me a sense of who tends to terminate in 2 and 3.
Coniocellular.
Coniocellular. So coniocellular deals with blue light. Well, who does the blue light want to get kind of married to, or at least be friends with?
Maybe red and green light.
Red and green. So tell me the two types that would be at 4A.
Parvo.
Parvocellular pathway. Give me the ganglion cell. Who's the
Oh, the midget.
midget? And then also, what's the other ganglion cell that could be here?
I mean, it could also be the parasol magnocellular.
It wouldn't be parasol. Why? Okay, so here's why I'm saying this, right? Because these two, the reason why 4A, I mean, this is just like a pedantic point, okay? Like I would have guessed that too also. I'm just saying the reason why, remember, 4C alpha is where you've got your, down here is where you've got your parasol cells. That's your magnocellular M type. But up here, we're getting married between the colors, right? Because it's close to here. This is where the colors are. So who carries colors red, green, which is the midget, and coniocellular, which is blue light, which is bistratified. Make sense?
Okay, yeah.
Right? And so those would be the two that I would expect kind of up here that would start to have shenanigans up here, right? Okay. As we approach this 2-3, if you're going to give me two, those are the two, right? Again, tell me what's down here. What is this layer?
The 4C beta.
You got it. And what ganglion cell then? It's the beta ganglion.
So the midget type ganglion cell.
Midget type ganglion cell, which is red-green color vision. You got it. That's it. So can I just point out something? We have gotten to a point where I'm literally just sticking metaphorical needles in the primary visual cortex. And you're telling me what is being carried and what is, what type of information that is, which cells, which ganglion cells. You could probably, once we get to the ganglion cells, we can go backwards and start talking about where, if it's on, off, right? We can start talking about the type of bipolar cell. And if it's like a midget, bipolar cell, then we can even go to one cone. And I think that this is a very powerful tool. You're not going to remember, layer 4A, right, is going to be like both coniocellular or whatever, but you might remember 4C alpha, 4C4, layer 4. You might remember 2 and 3 cytochrome oxidase blobs for coniocellular, but 4C alpha and 4C beta, layer 4, that's the striate cortex. And then this is going to be a layup on your tests, right? So I really think that this is really cool. So anyway, so that's that. This is a relatively quick one just because we know a lot already. You may not believe that, but we know a lot. So next time we're going to go back into the retina. Okay.
Back to the retina. Okay.
All right. Cool. Cool. Okay. So I will then see you then, and I will send you my handout in a bit. Okay.
Okay. Thank you, Dr. D.
No problem. Okay. Bye.
Bye.